Hello, my name is Vaughn Craddock, and I'm a medical student at the University of Kansas School of Medicine. I work in Dr. Navneet Dhillon's lab within the Division of Pulmonary Critical Care and Sleep Medicine at KU Medical Center in Kansas City. And myself, Dr. Cook, who's a pulmonologist here in Kansas City, and Dr. Dhillon have recently authored this review where we explore the role of extracellular vesicles in COVID-19 pathogenesis and as well as therapeutics in COVID-19. Much of our review can be summarized with this figure that we've created. So here you're looking at a alveolar capillary barrier with the end of the airway right here, the alveolus, and the capillary surrounding it, this vessel. So I'll go through the high points of this figure in relation to COVID-19 pathogenesis and the role that extracellular vesicles play in that, as well as the role of EVs in COVID-19 therapeutics. Extracellular vesicles are nano-sized particles that are important for intercellular communication. Their cargo might include proteins or nucleic acids, or they can also present signaling molecules on the surface of their lipid layer. In COVID-19, EVs can contribute to inflammation, endothelial dysfunction, coagulopathy, or dissemination of the SARS-CoV-2 virus. One of the important manifestations that can occur in COVID-19 patients is a hyperinflammatory response that's been colloquially named the cytokine storm. EVs can contribute to this by carrying pro-inflammatory cytokines, which are not only important for immune activation, but can also increase the permeability of the alveolar capillary barrier, which not only allows for molecules or virus or cells to enter into the bloodstream, but it can also increase the accumulation of fluid within the alveolar space. In other models of sepsis models that have looked at ARDS, researchers have found that there's a crosstalk that occurs between alveolar epithelial cells and resident alveolar macrophages. And this crosstalk is mediated by extracellular vesicles that carry nucleic acids like microRNAs that activate the inflammasome, or that can also alter the expression of tight junction proteins and contribute to increased permeability of the alveolar capillary barrier in ARDS. Lastly, there's a high degree of apoptosis that occurs within cells within the alveolar space. Some of this might be due to infection by SARS-CoV-2, but extracellular vesicles might also be contributing to this because they've been shown in other conditions to express the Fas ligand on their membranes and induce apoptosis. And this might be particularly important to consider in COVID-19 because lymphopenia is a marker of morbidity and mortality, and extracellular vesicles have been shown to express Fas ligands on their membranes and induce apoptosis of T lymphocytes. EVs from COVID-19 patients have been shown to cause endothelial dysfunction, as treatment of endothelial cells with COVID-19 EVs are shown to induce NLRP3 and caspase-mediated apoptosis, and have also been shown to activate endothelial cells to produce more cell adhesion molecules and also induce a more procoagulable state in these cells. This brings us to coagulopathy. So EVs that carry pro-inflammatory cytokines can increase the expression of tissue factor on the surface of endothelial cells and monocytes, but EVs themselves can also carry tissue factor on their surface and initiate clotting. We found in our recent study from 2021 that was published in Journal of Extracellular Vesicles that extracellular vesicle-associated tissue factor was higher in patients with more severe disease, and this significantly correlated with disease outcomes like severe clotting incidents or even mortality. Other studies have also shown that platelet EVs are involved in coagulopathy in COVID-19 patients, as those with severe disease have lower platelet EVs, and this is likely due to consumption of these EVs for clotting. And lastly, SARS-CoV-2 might utilize extracellular vesicles for dissemination of the virus. Other viruses like HIV or cytomegalovirus have been shown to utilize EVs for viral dissemination and also altering the immune response. Early studies have shown that EVs can carry the spike protein as well as viral RNA, which might support or provide some supporting evidence for this hypothesis. Extracellular vesicles might also be utilized in the future for therapeutics in COVID-19. One group has previously shown that by engineering EVs to express ACE2, the receptor which is primarily responsible for SARS-CoV-2 entry into host cells, that these EVs were able to block infection of healthy cells. And we provided an example of what that might look like here in the figure. Another option might be mesenchymal stem cell derived EVs, which have been studied in other conditions for their ability to reduce inflammation and promote tissue regeneration. We discussed one clinical trial that demonstrated that administration of bone marrow derived mesenchymal stem cell EVs was able to increase patient survival and reduce the need for invasive oxygen support in COVID-19 patients. These mesenchymal stem cell derived EVs are non-proliferative, they're small in size, which increases their mobility throughout the bloodstream, and they don't express HLA, reducing the risk of rejection. But there are some areas that need to be expanded on in research involving mesenchymal stem cell derived EVs. Their effects are far-reaching. They, in addition to inflammation and tissue regeneration, they also have been shown to affect metabolism and angiogenesis, which might increase the wide range of side effects for people who might be administered these EVs. And also at this point in time, high volumes are required in order to reach the full effect of mesenchymal stem cell derived EVs. So that concludes this presentation. Thank you so much for your time and your attention.